Hi, my name's Paul. In the previous couple of talks, I introduced independent component analysis, also known as ICA, and showed how we can use ICA to clean up or pre-process single subject fMRI data. In this talk, I'm going to explain the other primary use of ICA when applied to fMRI data, which is multi-subject or group ICA, where we run an ICA analysis on resting state data from an entire group of subjects at once. And we do this to identify the components or networks of activity that are common to the entire group. In this talk, I'm going to introduce multi-subject ICA and also a technique called dual regression, which we can use to relate these group-level ICA components back to each individual subject, and therefore to compare these networks across our subjects. So to recap, in single subject ICA, we take an fMRI data set and we rearrange it into a two-dimensional matrix of space by time. We then use ICA to decompose this matrix into a set of spatial maps and corresponding time courses. The most common way to apply ICA at the group level is called temporal concatenation, where we literally concatenate the data from each subject together along the time dimension and run ICA on all of the data at once. And just as with single subject ICA, what we get out of this is a set of spatial maps and associated time courses. But this time, each spatial map and time course, each component, represents some signal which is common to the data from all of our subjects. There is another technique for running group ICA, which I will only mention briefly. This is called tensor ICA. In tensor ICA, we stack the data along a third dimension and then run ICA on this three-dimensional time by subject by space tensor matrix. What we get out of this is a set of spatial maps and time courses, just like in the other approach, but we also get a third matrix which contains the subject-specific contributions to each component. The main constraint of tensor ICA is that it assumes that the temporal pattern of activity is the same across all of our subjects. So tensor ICA can be applied to task fMRI, where all of the subjects performs the same task with the same timing. But tensor ICA doesn't work well for resting state fMRI data, where we are looking at spontaneous activity throughout the brain for each subject, and this is obviously not going to be aligned across all of our individuals. So for resting state fMRI data, we need to perform ICA using temporal concatenation. So just to explain temporal concatenation again, we take the data for each of our subjects and we rearrange the data for each subject into a 2D time by space matrix. We then concatenate those matrices along the time dimension. Basically, we pretend that we have one really big, long data set. Then we run ICA on that big matrix, just like in single subject ICA. And what we get out is a set of spatial maps and time courses which represent the signals which are common in the data across our entire group. And as an aside, you may have noticed that by concatenating the subject matrices along the time dimension, we are assuming that we have anatomical correspondence within every single voxel. And this is a necessity when running group ICA. Basically, in practical terms, this means that before we perform group ICA, we need to register and transform the fMRI data from each individual subject into a common space, such as the MNI 152. And once we've done this, we will have the same number of voxels for every subject, and we will have anatomical correspondence across those voxels. So we can then concatenate the data along the time dimension and analyze it all at once. So you may be asking, why are we doing this? Why don't we stick with what we learnt from single subject ICA? We could run ICA separately on the data for each subject and then analyze each component separately across the results for all of our subjects. The problem with this is one of correspondence. When we run ICA on different data sets, we have no guarantee that we will get the same set of components. ICA is very sensitive, and small differences in the data may result in a particular signal being decomposed as a single component from one data set, but being split into several components in another data set. And there is no unbiased way of matching up these components across different data sets, nor to identify the activity which is common to the group that we're working with. So instead of doing this, we perform group ICA. We run ICA on the entire group at once using temporal concatenation. And this gives us a set of group-level components, components which represent signal which is common across all of the subjects in our group. The problem then becomes, how do we take these group-level components and relate them back to each of our individuals? And this is where dual regression comes in, which I'll explain in a moment. These group-level components, or networks, which we call them, have been shown to be extremely reproducible across different groups of subjects. There is a well-established set of resting state networks which are nearly always present in any group ICA analysis. Some examples of these are the default mode network, which you've probably heard of before, and the dorsal attention network. And there are a set of canonical resting state networks which you can expect to see in almost every group ICA analysis. But before we can perform any sort of statistical analysis on these group-level resting state networks, for example, to ask a question like, how does the default mode network differ between our two groups? We first need to identify the subject-specific contribution to these group-level networks. And to calculate this subject-specific contributions, we can use a technique called dual regression. We essentially use dual regression to take a group-level component or network like this and to estimate the subject-specific contributions, which tell us how strongly that group-level network is present in each subject. Conversely, we could look at it as showing us how much the data from each subject contributed to the group-level component in the ICA decomposition. So we run dual regression separately for every subject in our analysis. And as the name suggests, it performs two regressions, which are called stages 1 and 2. The inputs to the first stage are the pre-processed fMRI data for one subject and the group ICA spatial maps, so the group-level component spatial maps. The first stage of dual regression is a spatial regression, where we regress the group ICA components onto the subject's fMRI data. This results in a subject-specific time series for each group ICA component. In the second stage of dual regression, we feed the subject-specific time courses from the first stage into a second temporal regression. And what we get out of this is a subject-specific spatial map for each group-level component. So with these two stages, we can go from having a set of group ICA components to having a set of subject-specific components for each of our subjects. So here's another way to look at how dual regression works. In the first stage, we have the subject-specific fMRI data, and we have the group ICA components, and we do a spatial regression to solve for the subject-specific time courses. Then in the second stage, we again start with the subject's fMRI data, but now we have the subject-specific time courses, and we perform a second temporal regression to solve for the subject-specific spatial maps. So thanks to dual regression, we now have an estimate for every one of our subjects of the contribution to the group ICA component. And now that we have a spatial map for every one of our subjects, we can look at how those component contributions vary across our subjects using standard voxel-wise statistical analysis. So dual regression comes as a script in FSL. It doesn't have a GUI, so it's only available through the command line. It performs both the first and second stage of dual regression for you, and it also runs a third stage, which is a group-level statistical analysis using the randomize tool, which performs non-parametric statistics. When you call dual regression, therefore, you need to pass it a design matrix and a set of contrasts, and these encode the group-level statistical analysis that you're interested in performing. For example, whether you're interested in looking at group mean contribution to the default mode network, or a two-sample or an unpaired t-test between two groups. So the dual regression script will perform dual regression, and then it will run your group-level statistical analysis on every single group ICA component, and it will give you t-statistic and p-value images which have been corrected for multiple comparisons. And this is referred to as the third stage of dual regression. So this group-level statistical analysis works in a similar way to how things work in a higher-level task fMRI analysis or in a structural VBM analysis. The statistical analysis is run separately for each group-level component, and we take all of the subject-specific spatial maps for each group-level component. We specify a design matrix which contains our subject groups, parameters that we're interested in, or confounds that we want to control for, and additionally the contrasts which specify the tests that we actually want to run. Then we run a GLM, the same as how it's performed in a task fMRI, and the result for each group-level ICA component or each group-level network would tell us where there was a significant differences in strength and shape in this network as defined by the contrasts. So in the example on this slide, we are encoding a t-test where each EV, or we can imagine that we're encoding a t-test where each EV represents one group, and if we had a contrast of 1 minus 1 applied to this design matrix, we would get results which tell us where there was a significant increase in strength of this specific component in our first group relative to our second. So just to reiterate, before we can perform dual regression, we need to set up a design matrix. And we can do this using the standard GLM tool, which comes as a part of FSL. So dual regression produces a lot of output files, and these output files are numbered according to the stage of dual regression which produced them. Remember, we have the first stage is a spatial regression, the second stage is the temporal regression, and then the third stage is the statistical analysis. So first of all, you will get a text file for every subject, which contains the subject-specific time courses for each group ICA component. This is the output of the first stage of dual regression. Next, for each subject, you will get a 4-dimensional image which contains the subject-specific spatial maps for every group ICA component. And this is the output of the second stage of dual regression. And you actually get this second stage twice. So you also get another 4D image for each group ICA component, which contains all of the subject-specific spatial maps for that component. So this is exactly the same data as the subject-specific images, but it's just been rearranged by a group ICA component. And this is done because it's these files which are used as the input for the third stage for the statistical analysis. And this is what you get from the third stage, the outputs of the statistical analysis, which for each group ICA component and for each contrast which you specified, you will get a t-statistic image and a p-value image, which has been corrected for multiple comparisons. And these are generated using non-parametric statistics via the randomized tool. And one important point to note here is that while these results have been corrected for multiple comparisons within or across the voxels within each group ICA component, they haven't been corrected across the group ICA components. And what this means is that if you're interested in the results for more than one group ICA network, then you should apply a further correction using something like Bonferroni correction across all of the components that you're interested in. If you're only interested in one specific component, such as the default mode network, then you don't need to worry about this. And one more point to note on this is that if you're only interested in one specific component or in a subset of group ICA components, you need to still run dual regression on all of your group level components. And this is because all of the components which you're not interested in will act as confounds, and they will act to explain some variance that you're not interested in. If you only perform dual regression on the group level components that you're interested in, then all of the variance in the data, which is explained by the components that you excluded, will end up in the error term of the GLM and will therefore reduce the power of your resulting statistical analysis. So it's very important to include all of the group ICA components when you perform dual regression. So there's one last topic to cover in this talk. So one of the most important inputs to dual regression is these group ICA components or resting state networks. These are effectively used as templates in this statistical analysis, very similar to how in a VBM analysis you use a average gray matter template which forms the basis for your analysis. So when you're creating these group ICA templates using group ICA, it's important to use all of your data. It might be tempting to perform the group ICA just on your control group because you might have a group of healthy controls and a group of patients of some sort of neurological disorder. The data from the healthy controls might produce really nice, clean ICA components that look perfect, and therefore you might be tempted to use just your controls to generate the group ICA components, which you will then use as templates. But what this would do it would introduce a bias in your results, because the data from your controls will be much more similar to the data to the group ICA templates, because you only used the controls to generate the template. So there is much likely to be a much better fit of your controls to the group template, and you could end up with completely spurious differences because of this bias. So it's important to use all of your data, to use a representative sample of the data from all of your groups in your analysis. Another option is to use a completely different set of subjects. You could perform group ICA on an independent set of subjects, and this has the advantage that it's much less likely to introduce a bias towards one of your groups in your own data set. One of the downsides of this approach is that it's unlikely that it will model some of the other types of artifacts which will be present in your own data. Even at the group level, even if you have performed single subject ICA to pre-process and to clean up all of your fMRI data from each of your subjects, at the group level you will still end up getting some noise components or group level components which represent noise, which is common to all of the subjects in your group, and just may not have been strong enough at the single subject level to manifest itself as a separate component. And if you use an independent group, you will have the disadvantage that you're not modeling this group level noise. Another option is that in order to perform a resting state network analysis, you don't actually need to perform group ICA at all. You could choose to use a pre-existing group resting state network template, such as the one which is available through this link. This has the advantage that the results which you get may be more comparable to other results in the literature, but in the same way as using an independent control group, this has a disadvantage that these resting state network templates will not be modeling the noise or artifacts which are specific to your specific data set. In the end, though, there is no one correct method. It really depends on the type of study that you're running and the type of questions that you're interested in asking. The one thing that you need to do is make sure that you don't use a group template which is biased towards or against one of your groups. So hopefully after listening to this talk, you have an idea of how we can perform a whole brain analysis of resting state networks. We can use ICA to identify resting state networks of activity common to our group, or we can use an existing template of resting state networks as the basis for our analysis. We can feed those group level resting state networks into a dual regression to relate these networks back to our individual subjects. And then we can use standard voxel-wise statistical analysis to ask questions about how the spatial pattern or shape and strength of these networks varies across our groups. And I'll finish by mentioning this textbook. If you'd like to learn more about resting state fMRI analysis, then this is an excellent starting point. And you can get it from Oxford University Press or from Amazon by following these links, which are available in the PDF version of the slides. Thanks very much for listening.